though I missed most of the slide content. Now, starting our second day, just two small organizational announcements. We're going to have the poster session with the poster presenters present this afternoon, and before we'll have a poster pitch session where we have both in-person pitches and also recorded ones from the remote presenters. So I would ask all the poster presenters being here to line up according to the poster number when the poster pitch session starts. All your slides will be already in one row on the presentation PC, and then we can go in a blitz-like fashion, one after the other. Then tonight we're going to have dinner and drinks again, luckily, because I missed yesterday. Different location, different style, but I'm sure it's going to be great again. And so this time we have like a cap of roughly 3 drinks per person that is included, and afterwards one would need to pay for oneself. Because the dinner is a bit more expensive at that location than at the other. I just see that remote audience can't see me, so I'm just moving this a bit. Oh no, it's that camera. Okay, and that was it. No further organizational issues. So then I would invite Ellie Martin to chair the first session.
Thanks, Till. Okay. Morning, everyone. Thanks for joining us for this session, which is a kind of combination of neuroscience and technical topics. So we're going to get started with Kim Butts-Pauley from Stanford. He's going to talk about considerations for pulse shaping.
Thanks so much, and thanks so much for making it so early this morning. I did want to point out that this is some work that is collaboration between my lab and Leonard Verhagen's lab, and the work that was done at the Donders this summer when I was there on sabbatical. I'm going to go over some similar concepts to some of the things that we saw yesterday. In particular, what I'm going to talk about is sort of the need to disentangle direct and indirect effects, and particularly we want to maximize our efficacy while minimizing auditory confound, and then sort of bringing those together. What are the considerations for pulse design that might do that? And let's see if I can advance my slide. Okay, great. With respect to the auditory confound, this is something that has been known for a long time. So Gavriloff actually did this way back in the day. This is some figure that was republished in English recently. And what he was showing is that if you have a waveform in which there is an amplitude modulation that's a cosine amplitude, erased cosine here, that you can, as you change the frequency, obtain an audiogram, which you see over here. So just to review how that works. If you have air conducted sound, you have a sound wave that comes into the ear. It hits the eardrum, goes through those 3 little bones, hits the cochlea, which is laid out tonotopically. So the way you acquire an audiogram is you play a specific frequency. You keep increasing the frequency until you find the threshold, and then you can for which they first detected, and then you can draw a dot on the figure, and then increase your frequency and sweep through. And of course, humans are very sensitive to frequencies for air conducted sound between 32 hertz and 16 kilohertz. Ultrasound is defined as frequencies that are above that for air conducted sound. But of course, as we know, even using fundamental frequencies that are way above the hearing range, humans can still hear that, much as Gavriloff pointed out in his early publication, here showing with a fundamental frequency of 1 megahertz that it was clearly audible when he had a modulating envelope of different frequencies within the hearing range. Okay, so it's well known that bone conducted, or I'll also call it tissue conducted sound, has many mechanisms. So it can come in through the external auditory canal, through the bones, and then there's multiple different ways that sound can come in through the soft tissues, and those are well described in the literature under bone conducted sound. These are just some nice pictures. I like to look at these. They're very pretty. You can see the cochlea out here and some cutting through the vestibular organ there, and you can see the internal auditory canal here on an MRI bone picture. And I'll talk about MRI skull imaging a little bit later as well. Okay, so still sort of as a backdrop here, we did some prior work looking at auditory confound. I'll go over this very quickly because you may be familiar with it. Just very simply, we looked at an auditory brainstem response in rodents. It did include 3 different groups of mice, wild-type hearing mice, and 2 different strains of genetically deaf mice. And then we applied ultrasound as well as sound, and we had 2 measurements, the ABR and the EMG. And we applied different waveforms. So if you look at this waveform here, which is a rectangular waveform, then when we look at the power spectrum, which I've normalized, you can see here for the human hearing range from 32 hertz to 16 kilohertz, there are frequency components across that whole hearing range. When we considered the mouse in that prior work, this is the mouse hearing range, and there are large frequency components there as well. And comparing to the smoothed waveform or the ramped waveform that we looked at before, you can see that it was much less audible. It was down about 50 dB in the mouse hearing range. And so what we found was, of course, that that rectangular waveform, which has all these corners right here, was audible to mice, whereas the smooth waveform did not. Now, just to sort of rebut something that was said earlier, you don't have to have a waveform that has a frequency that is in the hearing range, but all of the corners themselves have frequency components that are across that hearing range. So even with longer pulses, if you have square or rectangular envelopes, they can still be audible. All right, so now moving on to new material. Since then, Zhihai Qiu, when he was in my lab, set up a system because we wanted to change out our readout from the EMG to something that was a little bit more of a direct readout. So he set up a GCamp imaging system such that we had GCamp modified mice. He had a ring transducer, as you can see here, and he cleared the skull so that we could get the optics that were through the center of the ring transducer. And unlike this prior work by the Caltech group, we had some differences in our study from theirs. In particular, we went normal to the surface. We had a different GCamp strain than they had. I don't really know if that made a difference. The one that we used had more fluorescence overall. And we did use the smoothed or ramped waveform, as you can see there. And lastly, our anesthesia was a little bit different, and I will mention anesthesia a little bit later in the talk as well. Okay, so here's a movie showing one of the responses, and there you just saw very briefly the ultrasound went on, and then we see is there at the focus that we see the fluorescence change at the focus. So that was really nice to see that it occurs right at the focus, and in fact, as we moved the ultrasound around in the brain, you can see that the fluorescence moves as we move it around as well. So that was a really good data point that it was due to the ultrasound itself and not due to the auditory effects. So we then wanted to quantitate this a little bit more. We put a region of interest here at the focus, and we over a number of mice. So here now averaging across 6 mice. And then we also put a region of interest over here at the auditory cortex. These are the curves that we see. And wait a minute, didn't I just tell you that these animals lacked ABR when we used a smooth waveform? That means that the peripheral auditory system was not involved. Given that the peripheral auditory system is not involved, why did we in fact see anything here at the auditory cortex? And I haven't fully resolved that. I think in fact that it's very likely that there's projections from other parts of the brain. But I will tell you the data that I do have. If we look over here at visual cortex, which is about a similar distance from the peak pressure point, then you can see that there's also a response at the visual cortex. These are just the individual traces in case you wanted to take a look at those. But the key point here is that when we're asking the question about whether it is causative, whether the auditory confound is causative for what we're seeing at the focus, that we looked at the latency to the 0.5% change in fluorescence. And we see that that latency is much lower at the focus and it's longer at the auditory cortex. And therefore, our conclusion was that it really can't be causative. The fluorescence moves with the focus, and it has the auditory cortex has a longer latency, and you can see that for the visual cortex having a longer latency as well. Okay, so that's great. Given what we think is going on with mice, again, that the peripheral auditory system is not involved when we use those smooth waveforms, we wanted to do a pilot study in humans. And so this is some work that Ben Kopp and Leonard Verhagen's group has helped with this summer when I was there. And what we did was we looked at two different waveforms, as you can see here. So these are 20 milliseconds long. One was a rectangular envelope and one was a ramped envelope with a 2-key window on the pressure, which is basically just a cosine there. We look at the power spectrum. You can see that the rectangular envelope has components across the whole hearing range from 32 hertz to 16 kilohertz. And the smooth waveform is down by 50 dB at those frequencies that humans are most sensitive to. So that's about 1 kilohertz to about 4 kilohertz. And you can see that it's down by about 50 dB. All right, so so one thing I wanted to point out is that this ramping and the response that we see in humans is not trial and error, but in fact, it's very clear how to reduce the components within the hearing range here. So this is the results that we got in this pilot study. So there were 7 subjects. And what Ben did was he modified, he had these 2 waveforms, which he applied intermittently with different amplitudes, different pressures. And he asked the subjects when they could hear them. And so then he came up with the threshold for each of those, and that's plotted here. Sorry about that, let me go back. And so with a rectangular waveform, about 20.23 megapascal was the threshold. And then with the smooth waveform, the threshold increased by 4-fold to 0.96 megapascal. All right, so it's very clear that the smooth waveform is having a very good effect on reducing the audibility. There's a number of questions still, and interesting points to mention here before I move on. One of them is you're probably wondering, well, what about the variability here? Is there variability in the subjects? And 6 of the 7 subjects were tested for their hearing, both with air-conducted sound and both bone-conducted sound, a regular auditory exam, and they had perfectly normal hearing. And so that's one thing to point out. Another thing to point out is that it's actually, the effect is a bit more subtle than this would imply, because what this implies is that these frequency components, so let me just back up a moment. This waveform right here, it has frequency components across the spectrum. It sounds like it's a very complex sound, more like a because it was played 3 times very quickly. And this one over here had, within this hearing range, didn't have any frequency components or very much. We're not very sensitive to this peak over here, but it did end up with a very high pitched sound, which is what these subjects reported. And so the variability here is sort of the variability in the subjects in perceiving that high pitch. So it's actually very interesting as to where does that high pitch come from? And back in the bone conducted literature, there's a lot often discussion about when people hear bone conducted sound, that it is in fact the highest pitch that their ear can perceive. So there's 2 components, 2 things that are going on, the envelope as well as that high pitch. All right, so that's where we are with respect to auditory confound and thinking forward as to how to improve that going forward, and especially like how are we going to do that if we want to have be able to interrogate duty cycle and et cetera. So there'll be some finessing of the waveforms for that. But let me move now to talk a little bit about efficacy so that I can then come back and put these 2 together to talk about how do we maximize efficacy while minimizing the auditory confound. So this gives the setup that I showed you here already. So this is our GCamp imaging, putting a region of interest there at the peak. Now, what Jurhai did is that he changed the pulse duration as well as changing the pressure, the peak pressure. And so, and this is the data that he acquired. So there's just with the higher pressure, he acquired longer durations. And what you see is that the fluorescence changes sort of flattens out here with longer durations. And so what it means to me is that at some point around 200 milliseconds, we sort of had the effect that we're going to have. And so the extra duration isn't really contributing anything. So just getting rid of those 2 points out there and only including these points out to about 200 milliseconds, what we see is that there's an increase with pressure and there's also an increase with pulse duration. All right, so we actually had more data than just that peak point. So including rings and what the pressure was in each of those rings and putting all that data together, plotting the fluorescence here as a function of pressure times duration. And so we get a curve that looks like this with a reasonable correlation, but I was just playing around with the data a little bit. I was on the train playing in Excel, and what did I come up with? Well, if I actually changed it to pressure times the square root of duration, then I have a much better fit. And so that was really interesting to me, but on the other hand, does that make sense? And so let me digress for a moment and talk a little bit about my thoughts here on the biophysical effect. And so, so there's a number of possibilities, and we've had discussions about this yesterday as the different possibilities. And I think that as a field, we're definitely moving towards mechanical, thinking a lot about mechanical sensitive ion channels. But I want to just sort of back up to this thought that Scott Hansen at the Scripps Institute has been talking about, in that it's really the lipid membrane, which is a sensing force. And so this is just from a review article talking about the fact that lipid membranes have different types of lipids in them. And as we increase pressure, then they self-organize into ordered domains and disordered domains. And these are a little bit more solid, such that you have molecules that reside in these lipid rafts. And then under mechanical stress or under ultrasound, in fact, what happens is that, I'm sorry about that, that the molecules that reside in these lipid rafts, when the lipid rafts become a little bit more fluid, that these molecules can come out, and there's this whole chain of events that occur that ends up in the activation of stretch sensitive ion channels. Okay, so given that that's a theory for ultrasound stimulation that really resonates with me. But now coming back to the work that we saw, that dependence on pressure and the square root of duration, that now I'm going to just hypothesize a little bit that in some ways it makes sense to me. So if what we're seeing then is a return with the lipid rafts are becoming a little bit more fluid, then that's the case where it's sort of decreasing pressure. And that when we see their fluorescence change, that it's the highest fluorescence change where there's the peak pressure, the highest pressure, or in fact, the peak rarefaction pressure as well is occurring there. And so one reason why that makes more sense to me than to think about strain as a mechanism is that if it was strain, you would really see the maximum, the highest fluorescence at the maximum strain, which would be more like a donut around the peak. And that's not what we see. We see the highest strain at the highest peak pressure. Okay, so one other thought before I move on is this why low frequency is thought to be more effective? And again, this is just, I'm going out on a limb and just sort of postulating here. And one of the thoughts here is that what we have is if this is decreasing pressure and then increasing pressure pushes us back there, that these two parts of the ultrasound are not necessarily the same. I mean, we see that with microbubbles, that these two phases are not the same, and you have sort of a cumulative effect. And in fact, if you think about, well, the time constant for this to occur and the time constant for it to go back again, it may not be the same. And so we may end up with sort of a cumulative effect moving over here, but that may also explain that you have sort of this push back into the order domains, why it's not potentially related to duration itself, but the square root of duration. So a weaker dependence on duration. And again, this is just me sort of thinking about this. I have no data on this, so take it with a grain of salt. Okay, so in terms of what pressure is effective, well, I mean, in the work that we did with our mice, it looks as if even going down to 0.16 megapascal was effective. That was, you can just see here these peaks in these curves. And of course, all of those pressures that we looked at were under the regulatory limits for MI and ISPPA, so down here in this regime. Okay, so one caveat about what I've talked about so far is that I've shown you these curves that we're looking at the GCAMP modification in all neurons. And these were pointing at the somatosensory cortex right here. And we saw these fluorescence curves. And in fact, right before Jurhai left for his faculty position at the University of Macau, he actually did the GCAMP modification in excitatory neurons only. And so we see something that looks quite a bit different. So there's a little bit of a peak here and then a big dip. So in fact, Keith Murphy did something that was actually quite similar. And so he had a setup that was very similar. He and Jurhai had talked together and came up with a similar setup where they had both of them ring transducers. Keith's was a little different in that instead of looking at the surface of the brain, he actually put a fiber down into the brain. And so a couple of differences there that, again, he was sensitive to GCAMP, but only a fewer number of neurons and not looking at the whole surface. Another difference is that because it was a smaller setup, he was able to have his animals under awake and moving around, while ours were anesthetized with isoflurane. But he saw was something very similar for looking at those excitatory neurons. So there's a little bit of a peak and then a big dip there. So that was really nice for me to see because it was similar results despite the fact that we had isoflurane and he had no anesthesia. But going back to his results, is really interesting to see that the inhibitory neurons had a big peak. And so that's probably showing, explaining what we're seeing over here with a big peak when we're looking at all neurons. So there's a big peak for inhibitory neurons and a small peak for the excitatory neurons, and then a dip here. And so our interpretation of this is that both sets of cells are excited, but the inhibitory neurons are shutting down the excitatory neurons. Keith actually put his fiber into different parts of the brain. So here it was the hippocampus, where he had both sets of neurons, as I just described in the last slide. He put it down here in the thalamus. The thalamus is mostly excitatory neurons. And again, we see the excitatory neurons are stimulated, as you can see here. And so that's nice, that also corroborating our theory here that the excitatory neurons are excited. And then lastly, down here in a different part of the brain, in the ventral tegmental area, which has more inhibitory neurons, and you see that those are stimulated as well. Okay, so now let's bring it back together. So what we said that we wanted to do was sort of maximize our efficacy. And then what we had shown was that we can have maximize our efficacy if we increase pressure, increase the square root of duration, while minimizing the auditory confounds. And so smoothing or ramping will let you do that. And it's, there's a mechanism for understanding how the smoothing will reduce those frequency components, is by looking at the Fourier transform of those curves. So when we use a smooth, then for the same audibility, we can increase where we are on the efficacy curve. We can increase the pulse duration and improve that where we are on the efficacy curve as well as staying under our limits. So that's why I think, given the data that we have, that my conclusion is if we can ramp our waveforms, and then, continuous wave seems to work really reasonably well and to have a good efficacy. That being said, I would like to go back and, use a pulse train and evaluate that as well and see if, in fact, it's any different from the continuous wave. All right, I am not ready to be done yet. I still have a few minutes. I have a captive audience. There's a couple more things I'd like to say. One of them is an additional thought here, and then I'll have one more after that. So one additional thought here is that, in fact, ultrasound stimulation can be quite loud. I mean, we heard some talks yesterday where people are saying, well, it's loud enough that we have to turn the masking up quite loud. So, and my experience is that it can be quite loud if you use a 1 kilohertz pulse train. It's incredibly loud, so much I sort of jumped away from the transducer. MRI scanners are quite loud. We know that's true. In fact, people have to wear earplugs. The combination is quite loud. Earplugs are required for MRI. Earplugs make bone conducted sound louder by 30 dB, which is quite significant. So my point here is that I think that we need to be concerned about this, about auditory safety. Being somebody who does have tinnitus, and probably because of all the MRI scanning that I had as that it's not something to see that. We really should keep an eye on this and pay attention to this and consider when we have normal subjects, what are the sound levels that we're applying it, and maybe even at some point think about safety considerations and limits here. One last thought before I conclude was a little bit about MR skull imaging. So we've seen some discussion about that yesterday. And I took a little bit of time this summer to just compare the ZTE on the GE scanner versus the Petra on the Siemens scanner. And of course, the third major vendor of MR equipment is Philips, and they also have an ultra short echo time bone scan. Just comparing these two, what you can see in the subject is very similar results. In case you're looking for trabecular bone, the subject actually didn't have very much trabecular bone, so don't worry about not being able to see it. The reason I wanted to show you this subject is that this subject does have calcifications in their brain. These are just a couple more images. These are the GE ZTE, again, 1 degree flip angle. And the 1 degree flip angle sort of maximizes the ability to see the bone versus the soft tissue. So you see beautiful depiction of the internal auditory canal. When you increase the flip angle, then you get a little bit more differentiation between bone and air, but sort of digressing into a point I didn't really intend to make. What I did want to do is just point out how beautiful these pictures are. And in fact, they're also quite beautiful at pointing out the calcifications that this subject had, that unbeknownst to the subject, who did not know that they had these calcifications. Okay, that's really important for us, because if we think about ultrasound that's going to go through the top of the head here and then encounter these calcifications, there's going to be reverberations and quite a bit of the distortion of the field. But it's more that I think that this could potentially be something that we need to think about in terms of safety, that calcifications could potentially be heating up as well and being closer to the brain even than the skull that's, actually in the brain. So it's probably something that we want to avoid. And of course, calcifications are something that the high intensity focused ultrasound field avoids quite diligently. So I have now said my two extra points that I wanted to make to my captive audience, and I will conclude now that ramping considerably reduces the auditory confounds, while in the work that we show, maintaining efficacy over the parameters that we did study, and long pulses are effective. Outstanding questions, how to do the optimal ramp. I just showed you a ramp that works nicely. It may not do everything that we want. And as I mentioned, there may be a pitch that remains, a high frequency pitch, but if we can understand what that pitch is, then that will make masking the residual perceptions quite a bit easier. And then of course, there's more to be done, looking at somatosensory effects, et cetera. So I would like us to keep in mind auditory safety and calcification safety. So just something to put in the back of your minds, it's something to think about. Okay, I really am going to conclude now. I'd like to acknowledge my lab group, as well as other faculty members that I work with, and of course, Leonard and Ben from the Donders Institute. And at that point, I'll stop and see if you have any questions. Do we have any questions? Do we have any questions from the audience? Yeah, can we get a microphone down here, please?
Thanks a lot, fascinating talk. I was wondering, what's the duration of your ramping? And when you compare pressure thresholds, does the ramping have any influence on the ISPTA?
So the way Jurhai did that with the ramping is that he had one waveform, and then we changed the duration of the pulse, then the whole thing increased in duration. So the ramping actually changed a little bit as he increased the duration. So a little hard to say exactly what it was, started maybe around 10 millisecond duration for the ramp. And the second question was about,
I mean, if the ramping is a significant portion of the waveform, it's going to influence the ISP, the average intensity. So I was wondering if you were compensating for that.
And so what I just showed you was that there was efficacy when I only was looking at the ramping, but what we do need to do is go back and say, sort of what's the fluorescence divided by sort of the pressure times duration, and sort of look at it from that metric, which we haven't done yet.
Thank you.
You see Leonard has a question.
All right, thanks. That's brilliant. Some people in the field, they're using slightly longer pulses of 5 milliseconds or 20 milliseconds at a relatively low pulse repetition frequency. But if we're going to look at some of the other posters, we're seeing that many people are using, 1,000 hertz pulse repetition frequency. Do you think there's any magic in these higher PRFs?
I'm the wrong person to ask if there's any magic, because I don't know if there's any magic. I'm trying to, I really scratch my head over that. And, because it just seems like if it's pressure times the square root of duration, then just more ultrasound energy is going to give us, more fluorescence here is our readout. So I don't understand how oscillating back and forth changes that here with the mechanism that I have in my head. So I don't know what there is. Somebody has to convince me that there's something there. And I'm all open to hearing as to what that magic is. So sorry, I just want to hear what it is too.
And then so moving forward, you're also suggesting we're looking a little bit closer at these longer pulses and these longer ramps and even. Okay, well, I'm ready to get started. Sounds exciting.
Hi, thanks for the nice talk. So how was the focal size and the frequency you use, and also the anesthesia for the fluorescence experiment?
Yeah, okay, so the focal spot size was huge because the mouse brain is tiny. But as you can see, it was estimating about, like 2 millimeters across, which seems big in that tiny mouse brain. The frequency he used was on the order of 470 kilohertz. And then the last question was?
The anesthesia.
Oh, yeah. So he used isoflurane, and typically what my students do is they go at a reasonable level of isoflurane to get the animal all set up. And then they go through a period where they've lightened the isoflurane to 0.6 or so, and then wait till, there's kind of like a toe pinch and a response from the animal. And then they have to be really careful that the animal doesn't crawl out. So trying to just kind of go as light as possible with isoflurane.
Okay, and regarding the size of the beam that you see reflected on fluorescence, does it match the intensity or the pressure? Do you check for
The
that?
fluorescence, the fit there, I did fit to intensity and didn't have as good a fit as pressure.
No,
So,
but I mean the spatial profile. Like,
this,
yeah.
oh,
Like the size of the beam, does it match better just the pressure or the intensity? Because with intensity it's a bit smaller.
well, that's just a question of how you look at it, how you report it. So, so I'm not quite sure what you're asking.
Yeah, I mean, the size of the ultrasound beam, if you depict the intensity, because it's the square of the pressure, it's going to be smaller.
Sure, but that's just a question of how you look at it. And ultimately, when we took those regions of interest for the quantitation, we could have reported it as intensity or pressure. And in fact, we did the analysis with intensity as well as doing the analysis with pressure.
Okay.
So it doesn't make any difference other than the figure that I showed you would have looked different if I had plotted that figure for the scan tank measurement as intensity.
Okay,
Yeah.
thanks a lot.
I hope I answered that. Yeah, okay.
Okay, thank you, Kim. In the interest of time, let's move on. Okay, so can I invite our next speaker, Alessandro Bongiorni, to come up? He's going to talk about transcranial ultrasound modulation of distinct neurocognitive mechanisms for task engagement and speed accuracy trade-off strategy in non-human primates. That was a very long title.
Hi, everyone. Sorry for the long title. I'm very happy to be here. Just recently moved to Paris, but I present this work that was run in Oxford. And I was, there is also another, my previous work was also with macaques and ultrasound, and I presented it as a poster at the previous FUN conference. So I'm happy to be here for the follow-up. And this is not yet published. So we are going to talk about speed accuracy trade-off. It's really a talk about decision-making experiments, but as you will see, we use ultrasound to probe how these neural mechanisms differentially affect behavior. So we start by explaining to you what I mean by speed accuracy trade-off, show first the general results in terms of behavior and fMRI data, and then go on. So bear with me until the second half of the talk to hear about the TOS experiment. Right, so I suppose everyone knows what is speed accuracy trade-off, but to make it clear, when you take a decision, you can sort of pay more attention to it, so slow down in order to make sure that your decision is correct, or you can sort of do it a bit more in a rush. So on one extreme of the spectrum, you have the strategy of the CEO here in the cartoon, who's saying, of course, we can make fast decisions once we have considered the 4,872 factors. So essentially, it's going to be a very long meeting before they manage to take this important decision. On the other extreme of the spectrum, you can have this Formula 1 pilot who's saying, if everything is under control, you're going too slow. So he's really trying to sort of push up things and may disregard small errors, for example, for this. Now, why we should ever make a trade-off between speed and accuracy? Because we need to accumulate the evidence. So in order to make the decision, you need to take information that can be external information or introspective information about your own preferences. And so there is an opportunity cost, because if you spend too much time doing this, you may not be able to do something else. For example, my third example is when I try to choose a movie on Netflix. If I spend way too much time choosing the movie and sort of looking at all the trailers, I will not have enough time to actually watch the movie. And animals know this very well. For example, monkeys foraging in the wild, because the opportunities depend on the environment where you live. So if you live in a rich environment, it means that when you miss out on one specific opportunity, you will have another next. But when your environment is poor, you may need to change strategy because you may not have other opportunities around. So this means that every time we make a decision, we can sort of move along this axis, whereby if you spend more time, we are more accurate. If you spend less time, we are less accurate. But you can also probably know by personal experience is that when you are particularly passionate about something and you spend a lot of sort of concentration on it, you can become both faster and more accurate. So you sort of break this trade-off. And if instead, like maybe all of us tomorrow evening will be very tired after a long conference, we may become inefficient, so both slower and less accurate when we are sort of making any type of performance. So what I want to see is essentially how animals, in this case, monkeys, sort of behave in a binary decision task in these two respects, and especially why they change their behavior. So in which conditions they will be higher or lower in the two dimensions. This is the task. So essentially is a binary decision. They see some cues on the left and on the right in terms of dots that appear one by one. And then they make decision, receive a feedback. So everything is very standard. They receive fruit juice that obviously they like if they make a correct response. And then what I vary is the amount of rewarded state that is skewed by the color and the frequency of the trials that represent a rich environment with many trials or a poor environment with fewer trials. And importantly, the duration of the sessions is fixed. So it means that they have always 40 minutes. If they spend more time doing one choice, they will have fewer choices in total. And I got 3 rhesus macaques for this experiment. We did a large number of sessions, so about 24 fMRI sessions, and then years later on a separate experiment, 24 TUS sessions. All right, so just to finish with the task explanation, I would like you to take the position of the subject and tell me whether there are more dots on the left or on the right. So the dots are going to appear one by one. And where you are sufficiently confident about which side is correct, you can raise your hand if you want. It's going fast. So that's it. I see some hands raised. So the left side is obviously correct. It takes a bit of sort of reaction, and there is a sort of risk to be taken whether you respond quick or slow. And as you can see, the subjects do the trade-off that I introduced, because when they are very fast, they are less accurate. When they are more slow, they are more accurate. Each animal has also a different profile, but all of them, they tend to reach a ceiling so that being particularly slow doesn't really help. Or if anything, the very slow trials are even worse, probably because it's when they lost concentration. So now, what the main purpose of my experiment was more psychological, was to study why they change their behavior in terms of SAT and in terms of efficiency. And so here are the results, but I will go quick on this because it's not necessarily related to ultrasound. But what we found, for example, is that the 3 animals have different strategies in what refers to stakes. So one animal becomes more cautious when the stakes are high, and the other animal becomes less cautious because it's probably more impulsive, which is probably not a good strategy, but it's what it is. And all 3 of them become less cautious when there are the frequency is high. So when there are lots of opportunities in the environment, they become less cautious. And you can see, for example, here that the effect of time for 2 animals is important. So toward the end of the session, they become more cautious. And for 2 of them, there is an autocorrelation, meaning that cautiousness in trial T is correlated with cautiousness in trial T+1. And then orthogonal to this, we have variations of efficiency. So for example, state make all of them more efficient, which makes sense. I was surprised to see that past rewards in the previous trial does not really make them more efficient, for example. And there is a negative effect of time. So as you probably know, by the end of the day, everyone is less efficient. All right. So then we have some neural results. The classic ones that confirm what we already know about decision-making systems in the brain are that if we look at correlates the reaction time during the actual decision, we can see classic motor and premotor areas, including frontal eye fields and cingulate cortex, which are not surprising. We can also see that if we correlate activity in the brain with the amount of evidence accumulated, which is a sort of proxy for accuracy, we see effects, well, in the motor areas, again, because they expect to suck up the juice, but also negative effect in VMPFC, which is consistent with previous results. Now, what I found most interesting to look at is instead not what happens during the decision, but what happens before the decision. So here we're looking several seconds in advance, and we want to see what neural activity predicts future cautiousness and what neural activity predicts future efficiency. So for the speed accuracy trade-off, the cautiousness, we find an effect in frontal pole. So this means that this area is going to determine whether the animal in the next trial will adopt a strategy that is faster as opposed to slower and more accurate. And the orthogonal regressor correlates with effects in subcortical areas. So it's nice to distinguish these things because the SAT strategy is probably more explicit and is done by the frontal cortex, whereas this is most correlated with area that may be linked to arousal, motivation, and reward processing, such as the habenula and the thalamus. So now, after this whole long introduction, I'm going to say what we did with ultrasound. So we identified these three areas that seem to affect not the actual performance of the decision, but the way you change your strategy and you prepare to make the decision. And so we differentially stimulated one of the three areas in separate sessions. So this is an across session design where each subject performed 24 sessions in total. And in each session, we disrupted offline the area, meaning that there was a single stimulation at the beginning for about 40 seconds. Then the animal was moved into the testing room, and then it was performing the task as usual for 40 minutes. All right. So yeah, usually I skip on these details for other types of audiences, but here we'll show. So we use 500 kilohertz stimulation to reach deep in the brain with a relatively small area of stimulation, because we were particularly interested in the habenula, which is a very small structure that, as you see here, is very clearly activated in fMRI. So we hope that by crossing 20 seconds for each side of the brain, the area of sort of crossing of the two effects should be sufficiently small to impact differentially the habenula from the rest of the surrounding tissue, in particular the thalamus. And we used 300 milliseconds burst every 100 milliseconds, so which is sort of classic paradigm that has already been used in our lab, Matthew Rashford's lab in Oxford, and also I think was developed or is similar to what the one was developed by Jean-François Aupé in Paris. And then for frontal pole, we use as a lower frequency transducer, mostly because it was an area difficult to reach without producing a lot of reflection on the skull. But we still managed, according to the simulation, to reach the target area with the same pattern of stimulation. Now, in terms of results, the first thing we saw, and that it was not necessarily what we expected, is that stimulating the thalamus made them significantly less cautious. So if you just look at the average behavior over the whole session, we expected more results from the habenula, for example, or the frontal pole, but it was really the thalamus that made them less cautious, means that essentially much faster in their responses, but obviously faster losing accuracy, and marginally more efficient. So this is not significant, but you can see the trend for thalamus making them more efficient. So this is one interesting result. Then the second one, that is consistent with what we predicted is that there was an effect in frontal pole for the autocorrelation of cautiousness. So here the idea is that you have this strategy. You can decide, now I'm going to be faster, now I'm going to be slower. And the strategy that you take in trial T is correlated with the strategy you were taking before, just because you tend to sort of repeat yourself. And this way of repeating your own pattern of behavior is increased after you disrupt the frontal pole. So here the idea is that if the region is important in order to change strategy, to adapt to the environment, if you disrupt it, then you stop adapting and you just keep repeating what you were doing before. So this is what we expected. We also expected a similar effect for habenula and efficiency, which was not significant. There was a trend for thalamus. And then on a third type of analysis, I also looked at how this evolves with time. So I'm skipping the details because this is not maybe crucial today, but we were interested in how different types of regressors affect the behavior. And I distinguish internal regressors from external regressors. And I noticed that essentially time was the one that predicts most of behavior. So you can see that cautiousness was really going up during a session of 40 minutes and efficiency was going down. But now the interesting bit is that you can see that brain stimulation does affect the behavior. And I found it interesting that especially in the second half of the session is affecting behavior, meaning that even if the stimulation was done only once at the beginning, the psychological effects may develop with time and become more evident toward the end of the session when they may interact with other factors like fatigue or satiety, etc. And this is the same data replotted by removing, so as a difference between the condition and the sham. And the error bars are across sessions. So here I just ignore subjects and I take the error bars across sessions. And you can see that essentially frontal pole increases cautiousness toward the end of the session and thalamus decreases it. So this is the last slide. As I said, there are different regressors that predict the behavior. So some of them are rewarded stake or frequency of the trials. These are external regressors. And then others are correlated to the internal state of the animal. So for example, the correlation between cautiousness and trial T and T+1. And essentially what we see is that in the first half of the session, but not in the second, habenula shows the effects that we would expect. So the internal regressor impact increases after stimulation, and the external regressors decreases, although it is not completely significant after stimulation. So it means that the overall balance of the behavior is less affected by what happens outside you and more about just repeating yourself. And so putting things together, I've shown that from a cognitive point of view, we can differentiate when the animals perform a binary decision task, the speed accuracy trade-off from efficiency. From a neural point of view, using a correlation measure like fMRI, we can disentangle different neural structures associated to these strategies. And finally, with the intervention method like ultrasound, we can show how targeting different brain areas produces differences in the effects, how there is a temporal trend in these effects that are not instantaneous, and how different brain regions are differentially impacting the behavior and the cognitive aspects. So with this, I would like to thank my supervisor, Matthew Rashworth, and everyone else who helped in this project, and you for your interest.
Thank you, Alejandro. We've got time for one very quick question. There is one. Yeah, can we get a microphone over there?
Thank you so much.
Hi.
It's very intriguing. Quick question, did you pre-register it? So,
Sorry?
did you pre-register your analysis?
No, it was not pre-registered.
Okay, thank you.
Yeah.
Thank you very much.
Thank you.
Okay, so let's move on to our next speaker, who's Benjamin Slater, who's going to talk about transcranial ultrasound stimulation in primates.
Yes, I just want to thank you for giving me the opportunity to present here. This is my first conference and of course, first time at FUN. It's been a fantastic experience so far. And today I'm going to provide some preliminary evidence that I've gathered during my PhD while at Newcastle University. It's still ongoing, so I'll be curious to hear your opinions on the data. So I'm going to be explaining how or what the effect of TUS has on the hippocampus and prefrontal cortex during context-guided sequence learning. So to provide a bit of background, both humans and animals utilize context every day. If you think about rats in a context sphere maze, they're able to associate what happened and when, and they remember this for days, months afterwards. And it's a good way to parcellate and separate different memories. And as such, context involves both the spatial and the temporal modality. Now, I've mentioned that rats, research has been done quite extensively in rats on context behavior, but less so in primates, for many reasons, one of which it's very difficult to modulate the external environment for a macaque. If you think about rats, you can change the maze, you can alter what's involved in the real world for them, but for macaques it's a lot more difficult because they're such a larger animal. So that was an aim of this task, was to see if we could modulate the wider contextual environment, as well as look at which regions are involved. And it's thought that there are two main areas. So the temporal lobe, namely the hippocampus, for processing these spatial and temporal elements, as well as the prefrontal cortex, which is potentially responsible for any conflicting memories that might occur between contexts and deciding what is relevant and when. So the overall aim was to use ultrasound to modulate these two different regions to see how they're involved in context-dependent memory. So regarding the regions, I selected the anterior and posterior hippocampus because there seems to be specialization at the two ends of the pole. Namely, anterior hippocampus might be involved in larger contextual environments, whereas the posterior hippocampus is more specific for objects and details. And then the medial prefrontal cortex, because this is the area that receives the densest number of connections from the hippocampus. So this will be the region that will be incorporating all the information it receives from the hippocampus. So we registered an MRI image of the macaque brain to BrainSight, which, as many of you know, is a frameless stereotaxic system. And this enabled us to then navigate the transducer against the skull so we could direct each region and just provide an exact location on repeated number of sessions. So once we'd established the right region, the parameters we used have been used previously in other macaque studies, and we used a frequency train of 30 millisecond bursts, each containing a frequency of 250 kilohertz, and the bursts were separated by 100 milliseconds, and overall stimulation lasted for 40 seconds. And each region was stimulated bilaterally per session. And I should point out at this point that we did both sham and stimulation sessions, and a sham session involved placing the transducer against the animal's head, but not actually providing any stimulation. They went through the same procedure of going to the lab, receiving all the setup, but they didn't actually receive the stimulation. So once we'd provided either sham or stimulation, the macaque was then brought back to its home cage, where it could interact with two touchscreens. So we got the animal to perform 5 trials on one touchscreen before then moving to the other, perform 5 trials, and then repeat this process through the session. And I'll explain why in a couple of minutes. And an example trial involved the animal first selecting a white spot on a screen, which would then present them with 4 objects. And dependent on which cage they were in, so whether it's left or right cage or the color of the screen, they had to select 2 objects in sequence. So for example, for this particular trial, they're in the left cage with a yellow screen, so they need to select objects A followed by B. And if they were correct, they would receive juice. And if they were incorrect and they made an incorrect choice, they would have to just abort the trial and move on. And we randomized the position of each of the objects across every trial, so as they couldn't learn a particular hand movement or a particular spatial location of the objects. They had to specifically identify the objects themselves. So for the initial learning phase, we created a global context. And by this, I mean they could use the background color of the screen, as well as the physical location of the screen. So as I've said, for the left-hand cage, it was always a yellow background. So they quickly learned that it was object A followed by B, whereas conversely, for the right-hand side, it was a blue screen. So it was object C followed by D. And what we saw when we stimulated the anterior hippocampus was that across a session, performance was generally improved compared to sham, with performance starting around 85% and dropping down as stimulation wore off across the 2-hour period of testing. Whereas with the sham session, they were generally worse, performing worse, around 75%, but then performance increased as they began to learn their sequences. So we saw an improvement in performance with anterior hippocampal stimulation, but with posterior hippocampal stimulation, we saw the opposite, with stimulation causing an impairment in performance, with performance generally being much lower than sham, with sham being around 95% on average across the session, correct? Whereas stimulation was around 90%, if not a bit lower. And then once the animal clearly learned the individual sequences dependent on the global context, we didn't see an effect for medial prefrontal stimulation. And this might be because the animal had already hit ceiling effect and was performing nearly 100% for every trial. So it might not have mattered for the stimulation. So once the animal had clearly learned the individual sequences, we then implemented a rule reversal. So what we mean by this is the sequences that were once relevant for the blue screen became relevant for the yellow screen and vice versa. So as you can see, AB was once only relevant for yellow, but now became relevant for blue. And ZD for blue became relevant for yellow. So this meant that the context hadn't changed themselves. It was still a yellow screen on the left-hand side that was static, but the sequences had swapped. So they had to re-update their understanding of what was relevant. And again, what we saw for anterior hippocampal stimulation was a vast improvement of performance across the session, with performance remaining quite stable around 90%, whereas for sham, it declined quite a bit, average around 75% correct. But for posterior hippocampal stimulation, we didn't see that drop in performance, suggesting maybe not particularly relevant for this part of the task, with sham and stimulation sessions having no main effect. But we did see an improvement in performance for medial prefrontal. They follow the same trend across the session, which might be indicative of maybe motivation towards the session, performance declining because of that. But they do show this improved performance with the blue line for stimulation, suggesting prefrontal cortex could be important for that rule reversal learning. And then finally, to make things quite a bit harder, we created a local context. So we removed the stability of the context color being dependent on the cage. So now the animal could experience the background color in any cage. So there was 4 types of trial, which I'll go through. You had on the left-hand side, a static context. So this is what they were used to, where the color of the screen remained the same throughout the trial. So if it was yellow, it would remain yellow, and they would select C and D as indicated. But for an unstable context, they would approach the screen, start the trial, and if it was yellow, they'd select object C, but then after that first object selection, it would switch to blue, and they'd have to re-update their decision and remember that as it's the second choice, they need to select the second element for that color. So it would be B, because they know that A and B are connected in the blue context. This was quite a lot harder for the macaque, but they showed good learning, although stimulation of anterior hippocampus showed no main effect on performance, with sham and stim averaging around the same across the session. But we did see the drop in performance, the impairment of performance with posterior hippocampal stimulation, with averaging around 65% compared to a sham of 75%. And then finally, we saw an improvement of performance again with medial prefrontal, with an average 85% compared to 75%. So it's indicating that maybe prefrontal cortex is more involved in this fast re-updating of decisions based on context. So to provide a quick summary of that, we saw improvement of the anterior hippocampus during initial learning, particularly when spatial location was important. So when we had that global context that was static, we saw great involvement of the anterior hippocampus. And then posterior hippocampus generally showed an impairment in performance, except during the rule learning, maybe suggesting that it's not relevant for sequence learning. They've already understood the relevant objects. So therefore, the posterior hippocampus is not involved. And then finally, medial prefrontal cortex improved performance, particularly during the local context when you had these fast approaching conflicting contexts. So when the update was required for the sequence, a fast pace. And for a direction forward, I'm currently training a second macaque to hopefully replicate these results, and we can see where we go from there. So it's some promising results. I'd like to thank my supervisory team at both Newcastle and Durham University, as well as the lab, because I couldn't have done it without them. Thank you.
Thank you very much, Benjamin. Right, do we have any questions from the audience? Yeah, there's one at the back. Can you get a mic then? Yeah.
Thank you for your talk. Would you go to for your first results now, please?
First results?
Yes.
Yes.
Yeah, yeah. Would you say it's an improvement in performance? Because it seems that the sham is decreasing with the simulation.
Well, which one, sorry?
So you mentioned that the anterior hippocampus improves performance.
Yeah. So generally across the session, you do see this decline in performance, but compared to sham across the whole session, performance was improved statistically.
So yeah, but anterior hippocampus doesn't improve performance.
Well, we saw an increase in performance with hippocampal stimulation. So
It doesn't increase across session, within a session, right?
no, it doesn't increase across the session. It's just,
Within a session.
yeah,
it
it's on
doesn't.
average higher.
I mean, one could argue that maybe it increases the alertness, just the sheer effect of the simulation.
Sorry, I couldn't hear that last bit.
So my point is that within a session, the blue curve doesn't increase.
No, I've plotted
So
it
how do you say it's improving performance?
because it's generally higher. So if you so the way I analyzed it was a three-way ANO, but I've just plotted it via time to show you the sort of the general trend.
Yeah, but it could be because of the very early try-ons, which they are significant. I mean, I don't know significantly, but they are different. And as time passes, they more and more equal. So one could argue that, I don't know, the sham conditions also like going up in the anterior hippocampus across time.
Yeah, so the sham does show a general learning effect because we did it as a block design. This was a caveat of this initial phase. So you see sham increase across the three
Exactly.
three regions, but for the latter two phases, we implemented a counterbalance randomized sham and then stimulation session. And the fact we see the similar trends of increased performance of anterior hippocampus makes me think that we do. I mean, I'm happy to chat about this further with my poster.
All right, thanks.
Thank you. So one question I have a lot for these kinds of experiments or the use of pulse ultrasound in general, and maybe for your case specifically. So we have a relatively small focus of the beam and a relatively long structure like the hippocampus. So you target anterior and posterior parts, but still, even within that anatomical structure, how do you decide where you target? And would it make a difference if you're a centimeter more anterior or centimeter like?
Yeah, it's a good question. I kept to the extremities. So the hippocampus is around 12 centimeters long and the width focal beam was, I think 4. So I kept it to the very extreme ends of anterior and posterior. But you are right, there could potentially be overlap, but try to avoid that, like I said, by keeping to the extremes.
Okay, what would you say, what's the percentage of the vocabulary volume that you covered roughly?
So a third, so 4, 12. So we keep to the, very end of it. Yeah.
Thank you.
Okay, thanks very much, Benjamin.
Thank you.
Let's move on to the next speaker. So the last speaker in this section is Hector Estrada, who's going to talk about fluorescence-guided transcranial focused ultrasound in the mouse brain.
Let's see if it works. Thanks. Yeah, okay. Hello to everyone. I'm glad to be here to show you some of our results. I would like to start by thanking my colleagues and co-authors and also to our funding agencies. So we have seen also today some very nice results, but using fluorescence to look at what happens in the mouse brain when you deliver ultrasound. And one of the issues that you already observe is that if you use very low frequencies, you cover a large volume of the mouse brain. And this makes difficult to interpret the results you get because, which region is producing the effects that you observe. So one way to get a tighter beam is actually to increase the frequency. That's like a well-known recipe. And you can see that if you use a linear array here in the red spot, if you increase the frequency up to 5 megahertz, you can drastically reduce the focal spot. But that's not the only way. And if you remember basic physics, if you increase the angular coverage, you can actually get even a tighter beam, as in our case, even if you use low frequency, like in this case, 3 megahertz. So, and we actually have this capacity in this kind of array. If you look here at the left, it's not that we designed this specifically for ultrasound neuromodulation. Actually, our lab focuses strongly on optoacoustic imaging. And for optoacoustic imaging, having this 512 channels spherical array is actually the best way to perform optoacoustic tomography. So we decided, okay, let's try to do ultrasound neuromodulation, and we started using fluorescence in GChem mice. So we use this fiberscope here, and although the aperture is very small, you can still cover the whole mouse cortex. You can see that the field of view is roughly 12 millimeters. And we characterize our array and our focus, and the first thing we tried was to measure the insertion loss of a mouse skull. And at 3 megahertz, you can see the losses are about 6 dB. And if we check the ultrasound focus, you can see that it's below 400 microns. And even going through the mouse skull, we can keep those dimensions, although you can see there are some shifts in the focal position. So we turn on the ultrasound knob, and then we observe something very weird. a decrease in fluorescence. Here you can see the typical readout that we have from this fluorescent measurement, and these very large oscillations are resting state activity. So we use 1.2% isoflurane as anesthesia, and we have very synchronized activity in the anesthetized mouse brain, which is a very strong background. But even then, we observe that when we turn on the ultrasound during these 150 milliseconds, there was a decrease in fluorescence that, at the very beginning, we didn't know what it was. We could see individual frames where this happened, and we had to actually develop some techniques to compensate for the very high background of the resting state. And because we have an spherical array, we can do three-dimensional beam steering, and we try to do that. And first, we point our ultrasound focus to the red circle, and then we change the delays, and we obtain this, all the focal spot at the green circle. So this is very cool, but we can spot where the focus is, but what it is. And it's just for quenching of the fluorescence because there is a thermal rise. We are using relatively high pressure in the range of a few megapascals. And we run an experiment on a slice of this GCAM mice, and we decrease the temperature. Sorry, we increase the temperature, and the relative brightness, fluorescent brightness, decreases. And this is further proven by fitting spatiotemporal thermal model that fits very well with the observations we have from fluorescence. So this is the average among many mice, I think more than 7 mice. And you can see how the fluorescence decreases, and then after you turn the ultrasound off, it goes back to the baseline. And actually, it has the size of the focus is slightly smaller, and this was my previous question to Kim, because this in this case fits the intensity, because the intensity is what produces the rise in temperature, and this is what we see reflected here in the fluorescent change. Unfortunately, we cannot use this to make absolute measurements of the temperature increase. That would be great, but as far as we went with this modeling, we can only tell relative changes, and we will need a calibration that I think is not very easy to perform in order to get a temperature readout from these negative fluorescent changes. Because we can observe nearly the whole brain cortex, we perform functional connectivity analysis. So we split the mouse brain in different compartments, and then we look at the Pearson correlation among them. And this is an example, for instance, for the baseline. And here you can see the same for 3 megapascals of pressure. So then you can form a matrix, and after 20 sonications at 3 megapascals, there is barely any difference here. But after one sonication at 3.9 megapascal, which is a very high pressure, we see a difference. And then with histology, we actually confirmed that for 3 megapascals, we couldn't see any sign of damage, whereas for one sonication at 3.9 megapascal, we see a very small but clear damage, which looks like a vessel that was burst. And how does it look when you deliver these high pressures into the mouse brain? So because of the damage, you create something that is called cortical spreading depression that has been shown yesterday, I think also by Gu from the Shapiro lab in Caltech. So you basically have a huge change in fluorescence in the order of 160%. or even more that travels only through one of the hemispheres of the mouse brain. And then you have a strong decrease that is not permanent, but it stays there for a few minutes. You can see that this wave propagates very slowly. You can see it lasts for 2 minutes. And we studied the dynamics, and we also tried to find what was the threshold that triggered these CSDs. So you can see here that for 3 megapascals of pressure, we tested in many mice, many sonications at 3 megapascal, and we never observed any CSD being produced. But as we increase just a tiny bit the pressure, we observe that sometimes we trigger this CSD, and if you increase the pressure even further to 4.5 megapascals, you always trigger this. And this is, so we calculated more or less the temperature increase that will lead to this, and agrees with the literature. I think it's in the range of 47 degrees. When you increase by that bit, you cross the threshold, and then you trigger this cortical spreading depression. Yeah, so this is what we can do with our setup, but we have seen here already very nice results on stimulation. So can you actually see activation using this setup? And the answer is yes. We can see local activation is robust. We tested in many mice. You can see actually here the localization of the dip of the thermal, of this fluorescent thermal tag. And then you can see that on a region that is larger than the ultrasound focus, slightly larger, it's not much larger. You can see the activation here, and here is the time trace of that activation. The activation is rather small. You can see that here the change is in the order of 1%. And yeah, you might ask why we only show just half of the brain here. And the reason is, if you remember previously, the resting state oscillations, this background is very large, and the oscillation there is much larger than 1%. So because there is a very high correlation between the 2 hemispheres, that the activity is very large, but also very synchronized. You can see it in the fluorescence very easily. We decided to subtract the fluorescent changes from one hemisphere into the other. But yeah, we can see also in the full brain when this activation happens, but it's not that obvious as if we subtract the activation on the 2 hemispheres. Yeah, so that's it for today. Thank you very much, and I hope I convince you that this setup is very valuable, not only to study the ultrasound safety. As you can see, we can observe directly in real time when we damage the brain, but it also has a high potential to study ultrasound neuromodulation. So thank you very much.
Thanks very much, Hector. Do we have any questions from the audience? Yeah, we've got one just here.
Can you like head bar them and have them running on a wheel? Are they anesthetized for this? I think it's an amazing tool, but if they're like really anesthetized, your effects are probably pretty pressed down, right? Are they freely behaving on a wheel or something? I mean, are they moving?
No, I mean, we didn't check if they're moving actually, but yeah, we have plans to upgrade this, and there is the possibility to perform these experiments also in awake mice with the head restrained, though, because yeah, you have seen the array is very big. We cannot do like moving animals with this array, but head restrained, yeah, that will be possible.
That sounds cool. And I got one more. I think the temperature you were verifying through simulation, right? Or is there experimental? Because I see that too, but my temperature is dropping. And I'm wondering if it's like blood moving away, which is highly autofluorescent. Have you looked at that or thought about it? I could be wrong.
Yeah, I mean, we kept the pulse very short, like 150 milliseconds, and because of that, we can only see the decrease, and the temperature model actually fits very well. But I can see how this could change if you use longer pulses. Yeah.
Yeah. Okay, sorry, can you switch them up?
Have you tried to use GFP only instead of GChem in order to sort of disentangle effects of neural activity from purely thermal effects?
Oh, yeah. I mean, we actually tested this in bacteria, just using GFP, and we saw the dip very clearly there. So that convinced us that it's just a thermal effect and it's not related with the neurons. Although, I mean, if you use longer pulses, you might affect blood flow as well. And then, yeah, you will have the combination of two, the temperature rise and then a change in fluorescence due to hemodynamic changes.
Thank you.
More questions? Right, I have a quick question.
Yeah.
The last slide, the neuronal activation, what pressure level were you using there?
Oh, this is 2.8 megapascal. It's relatively high pressure. And yeah, as you can see, we can also see the dips. So there is a rise in temperature as well.
Okay, thanks very much, Hector. Okay, so let's thank our speakers, and then we have coffee ready at the back. Thanks.